Allele ID offers comprehensive primer and probe design functionality for pathogen or bacterial identification, species or taxa discrimination, and cross-species research. You can design primers and probes for microarrays or for TaqMan, molecular beacon, or cybergreen qPCR assays. Better yet, use both the techniques. Verify microarray results using qPCR, or perhaps make microarrays based on probes proven through qPCR. Allele ID first aligns sequences using ClustalW. It then analyzes conserved and species-specific regions to design probes and primers that amplify only the species of interest from the mix. We have incorporated an innovative feature we call minimal set to identify more than one species using a single TaqMan assay. When selected, Allele ID designs the fewest number of primers and probes that will uniquely identify each of the desired species. We hope you will be pleased with the time, effort, and cost savings of this approach. The taxa-specific or cross-species assay designs a single probe and a primer pair to identify a taxa or a group of organisms. Allele ID limits the search to conserved regions. For challenging problems when such designs are not feasible, Allele ID minimizes mismatches, especially towards the more problematic 3' probe end. When the group or taxa is highly dissimilar, which makes both of these approaches impractical, the innovative minimal set option can be used to design a set of oligos that will identify the taxa. To ensure specific amplification and exact detection, PCR primers and oligos are automatically designed to avoid regions of significant cross-homologies. For selective cDNA amplification avoiding gDNA, Allele ID interprets GenBank sequence header annotations and designs primers across selected exon-exon junctions. With Allele ID, you can design cybergreen quantitative PCR amplification assays, dual-labeled fluorescent TaqMan, minor groove binding or MGB TaqMan, FRET assays popularized by certain cycle manufacturers, and molecular beacon assays. You can also design fluorescent probes for any of these assays that would be compatible with previously designed cybergreen primers. You can conduct SNP genotyping or gene expression studies. For single-tube multiplex reactions, you can even design oligosets by minimizing TM mismatches and cross-dimerization. Allele ID can design thousands of probes for making microarrays or thousands of PCR primer pairs for cDNA and splice variant microarrays. You can efficiently retrieve, analyze, and store data and export the results for ordering synthesis. When you open the program in demo mode, the demo project will load automatically. It contains sequences and results to demonstrate the features of this program. When the program is activated, you can work with your own sequences. The first time you open the program in activated mode, it will load the default project. You can start working with it or create a new project. You can open multiple sequences in the project directly from NTRES, the SNP database, dbSNP, or from files saved on your local drive. To retrieve sequences from NTRES, click the button on the toolbar, or go to File menu and select Open, Sequence from NTRES.
To open multiple sequences, you may type or paste lists of accession or GI numbers. To open a sequence from a file saved on your local drive, click the button, or go to the File menu and select Open, Sequence from File. Select one or more files using the standard keystrokes. For Windows, use Control-click to select multiple sequences and Shift-click to select Arrange. The files must be in GenBank or FASTA format and may contain one or more sequences. Your sequences will appear in the Sequence Information table and will be saved in the built-in database until you explicitly delete them. Sequences stored in unsupported formats can be easily converted to FASTA format. To convert, use a simple text editor such as Notepad. Type a greater than sign on the first line, followed by a sequence identifier. You can add a description on this line, but it is not required. On the next line, paste or type the sequence. Do the same for any additional sequences and save the newly created file. Alternatively, you can import a sequence from another application by cutting and pasting it into the New Sequence window. From the File menu, select New, Sequence. You can also edit any sequence using this window. The Sequence Identification table shows the accession number, a brief description, and length of the loaded sequences. It can be sorted by any column by clicking its header. The right frame contains a graphical view of the selected sequence. It shows the designed primers and probes. A base grouping of 3 or 10 bases may be selected in the menu, View per block. You can also view single or double strands by selecting View, Sequence Strands. To copy the sequence of the template, the primers, the probes, or the amplicon, just right-click in this area. Use the menu to copy what you want to the clipboard. You might, for example, want to paste it in your browser for ordering synthesis. We also have a utility we call the calculator in the Tools menu. You can use it to complement or reverse complement a sequence or to obtain its base count or GC%. We refer to species-specific and taxa discrimination or cross-species assays as related gene assays. Allele ID can design TACMAN, TACMAN MGB, CyberGreen, or molecular beacon-related gene assays. You can also design oligos for spotting on microarrays. Single template and multiplex assays are available for TACMAN, TACMAN MGB, and beacon chemistries. FretProbe and CyberGreen primer chemistries are also available for single templates. To design related gene assays, you can use the built-in ClustalW algorithm to align the sequences of interest. Alternatively, you can load pre-aligned sequences stored in a .aln file on your local drive. The strategy of finding and exploiting minor differences in gene sequences has become one of the most important techniques for species identification and allele or strain discrimination. Allele ID uses alignments to locate these differences and designs probes and primers to amplify and identify mismatched DNA segments. To ensure specificity, aligned sequences are blast searched. The results are interpreted to locate the primers and probes so as to avoid false priming. For instance, let's consider the bovine MHC sequences, which are a highly conserved set of genes. MHC typing is used extensively in tissue organo or genotyping. The program aligns the sequences, analyzes conserved and species-specific regions, then designs primers and probes to amplify and detect only the species of interest. Select the sequences BF1 to BF9. Click on the button, or select Analyze, Align. Give a name to the alignment and, if necessary, modify the ClustalW default parameters. Refer to EMBL or EBI documentation on any ClustalW mirror site. Then press the Align button. If you already have an alignment, you can import it. Click File, Open, Alignment Sequence. To work with alignments, you must first click the Aligned Sequences tab. Now click the Sequences view in the lower pane and the Full View button. The upper pane of the Graphical View tab depicts the entire set of sequences. The lower pane shows the base detail at your cursor position. The upper pane, the conserved regions are orange, moderately matched positions are green, moderately mismatched positions are black, and unique positions are blue by default. Asterisks in the bottom row indicate conserved positions of the alignment. Right-click anywhere in the graphic to change the zoom and colors. You are now ready to design species-specific pathogen detection, taxa-specific or cross-species assays using the alignment results. Let's start with the TACMAN assay by pressing the TACMAN button. Be sure the Aligned Sequences tab is selected, then go to Analyze, Related Gene Assay Design, Species-Specific Design. By default, all of the aligned sequences are in the Bind To box. The primers and probes will be designed to amplify and discriminate each of these sequences. If the mix contains sequences that you specifically do not want to amplify, highlight them and click Unselect. When you choose the probe type Unique, you are asking Allele ID to design probes to identify each sequence in the Bind To box, one probe per sequence. The program will search for base positions that are unique to each individual sequence in the alignment. The probes are centered as close as possible over these unique bases so that even when the mismatch is only one base, the assay succeeds. When you choose the Minimal Set option, Allele ID will design the smallest number of probes required to identify the sequences. The minimal set will generally contain fewer probes than the number of sequences you wish to identify, and a unique combination of probes will identify each sequence. More on this later. After designing the probes, Allele ID designs the primers to amplify the sequence around the probe with the desired probe primer spacing and product length. There are two choices available for primer design, Unique and Optimal. When you choose Unique, the program designs a primer pair to amplify each sequence. Each primer is completely homologous to its template. When you choose Optimal, the program minimizes the number of primers required to amplify all sequences. The goal is to minimize the synthesis and overall assay costs. Primers can bind to a sequence and amplify it even if they contain a limited number of mismatched bases, especially towards the 5' end of the primer. Taking advantage of this fact, Allele ID designs the fewest number of primers to amplify all of the sequences in the Bind To box. The Optimal design option is especially useful for sequences with low similarity. In the Output Options section, set the number of alternate probes you wish to design for each sequence. A large number is useful when you are working with a challenging assay, offering you a choice for manual selection. You can select up to 50 alternates. The TACMAN, Beacon, or CyberGreen Parameter tab lets you specify the assay design parameters. You can even use the primers designed in the CyberGreen mode. First, design a species-specific assay in the CyberGreen design mode. When you are ready to design the probes, check the option Use Primers designed in the CyberGreen mode. Allele ID imports the primers for each sequence in the alignment and designs compatible probes. Let's look at what is meant by unique, optimal, and minimal with a TACMAN example. First, we'll select Unique Probe and Unique Primers and click Search. Looking at the Multiple Sequence Alignment graphical view, we find that the probe for sequence BF4 is in a conserved region of the alignment, but contains a mismatched base in the middle, which is unique to this sequence. The flanking primers are also unique to this sequence. The highest rated primer probe set is listed in the Probe Details tab. If the program cannot design unique probes or primers, it reports the status as not found. Let us now select Unique Probes and Optimal Primers and click Search. The graphical view shows common primers for BF6, BF2, and BF5, whereas the probes are unique for each sequence. Let us now search for a minimal TACMAN set with optimal primers. The program designed a set of 7 probes to identify 9 sequences. The assay can be viewed by clicking the Probe Details in the Multiple Sequence Alignment view or clicking the Minimal Probe Matrix tab on the Properties pane. A plus sign in the Minimal Probe Matrix indicates that the probe detects the sequence. A Y in the status column indicates that a sequence can be uniquely identified by using one or more probes. In this example, sequences BF2, BF7, and BF8 are identified by a single probe, while the rest of the sequences are identified using a combination of probes. A taxon is a group of organisms which are different both phenotypically as well as genotypically, yet they show a degree of conservation in their gene sequences. Allele ID makes use of the conserved regions to design probes to identify which individuals are members of the group. Of course, the use of this functionality is not restricted to taxa identification, but can be used to amplify and detect any related group of genes or for developing cross-species assays. In a large number of organisms, 16S rRNA sequences are conserved. Such conserved regions can be used to design probes to categorize individuals into groups or taxa. This technique is useful in a number of areas, including biodiversity studies, classifying pathogens, bacteria, and diseases. For example, identifying hepatitis B from C. Let's demonstrate with the nematode 16S rRNA and start by aligning these 9 sequences. To design taxa-specific assays, be sure the Aligned Sequences tab is selected. Then go to Analyze, Related Gene Assay Design, Taxa-Specific Design, or click the button. Select the sequences to bind or to avoid. Selecting the conserved probe type restricts the probes to the conserved regions of the aligned sequences. Thus, only a single probe would be needed to discriminate each group or taxa. Let's look at the output of the conserved primer and probe design. If such a design is not possible, simply select Mismatch Tolerant. The goal of the mismatch tolerant assay is to design a single probe to discriminate a taxa. The probe may include up to the specified number of mismatched bases, which will be located as close as possible to the 5' end to maximize the probability that the probe will bind to all of the sequences. An option to output degenerate probes is also available for synthesis of a probe pool, which should improve assay success.